All right, welcome to this lecture on root anatomy and physiology. Throughout this video, we'll be discussing the anatomy of roots and so basically what the parts are and the physiology. How is it that roots accomplish what they accomplish for plants? Um, yeah, let's just jump right in. Okay, so to begin with, I hope we all know generally what roots are. I hope we've pulled up a weed or, maybe pulled out old garden plants a couple times in our lives, maybe dug up an unwanted shrub or tree and experienced roots. Roots are one of the five major plant organ systems, along with stems, leaves, flowers, and fruits. And because roots are typically found underground in most plants, they often go kind of unnoticed and unthought about. And I would like to challenge that throughout this video today. Roots, in my opinion, are one of the most important plant. I would say that they have an equal importance to the rest of the plant organ systems combined. Many plants can be cut down completely and resprout from roots. But if you cut the root off of a plant, they don't often survive. So I consider roots to be extremely important, and they really allow most of the processes above ground to occur because roots are how plants collect the raw materials they need for life. Okay. So roots have three basic functions. We already talked about how they collect materials that plant needs for life, but what are those materials? Um, Mainly water and dissolved mineral nutrient salts, which are found in their substrates. Roots also serve to anchor the plant to the substrate. Um, if you're not sure what a substrate is, that's just a material or surface that plants grow in or on. It does not have to be soil. This could be potting media, it could be the bark of a tree, it could be a rocky cliff face, it could be the body of any other plant. Um, Plants really have found ways to grow on pretty much any surface that is, reliably solid. Um, so I will probably not be referring to soils a lot in this unit. I will be referring mostly to substrates. Because in horticulture, we don't typically grow plants in soil unless we're in a landscape. In a nursery or in a greenhouse setting, we're typically using soil as potting medias, which are composed of mixtures of components like peat or bark chips or perlite or vermiculite, and those are not really soil components. So just to summarize, roots anchor the plant. They help the plant absorb water from the growing substrate, and they help absorb dissolved mineral nutrient salts from the substrate, the new mineral nutrients that plants need in order to live and grow. Um, I think that it's helpful to start this discussion about plant anatomy or root anatomy, I should say, with the longitudinal anatomy of a root, starting at kind of the tip of the root and then working our way back so that we can really understand how these roots are growing through the soil. Um, we'll be looking at this list of root parts um, as we go. Okay, so, like we have at the tips of stems, we also have apical meristem at the tips of roots. And the root apical meristem is essentially this region near the tip of the root where cells are actively dividing. Those cells divide very, very rapidly, and they don't really have time to grow bigger. They're just small cells that continuously divide. They're duplicating their DNA, they're duplicating all of their organelles, and they're just pinching in half and making new cells. Um, because the root apical meristem is composed of this soft, rapidly developing, rapidly dividing tissue, they require some protection as they push through the substrate. So the root apical meristem actually has a layer of cells which are being pushed forward towards the tip of the root, which will develop into the root cap. and roots that are being pushed backward that will develop into the kind of the permanent structures of the root. Um, just to remind you, if you haven't watched our lecture on stems yet, a meristem is any region of the plant where um, cell division is actively occurring and new plant tissues are produced. Okay, so this root cap is really, really interesting. So if we look at the apical meristem, it's composed of these very, very small, rapidly dividing cells. And they're building the root behind them, but they're also building the root cap ahead of them. And so there's actually cells moving kind of back and forward in this scenario. And as these root cap cells mature, they're pushed farther and farther out towards the tip of the root. And then they're eventually sloughed off as the root pushes through the soil. And you can think of this as kind of creating almost like a lubrication for the root tip as it moves through the soil. Roots are pushing past little bits of sand and grains of sand and pieces of gravel and soil particles that are very abrasive. And so the root needs to not only have kind of this tougher tissue that can push through that, but also a way to prevent damage to the root tip. So these root cap cells that have been pushed out from the apical meristem to the very tip of the root cap, they die and slough off, and that helps the root grow. The root cap, the living cells deeper inside the root cap, they actually contain there are some cells that are called stratocytes, which detect gravity, and that's one of the ways that roots can tell which way is which, and that helps them grow downward into the soil. Okay. Um, Once new cells have been produced in the meristematic region, um, they, the cells in the meristematic region in the root apical meristem are constantly dividing. So the constantly dividing cells are kind of always pushing some of the cells kind of back farther um, into the root. And in this region, which we call the region of elongation, those new cells which were produced by the root apical meristem, they inflate with water, and that's what causes the root to grow longer. So the elongation of the root in the region of elongation is actually pushing the root apical meristem down through the soil. So the plant is basically, by inflating its roots with water, that it's just shoving its apical meristems farther and farther down into the soil in search of water and mineral nutrients, and that's protected by the root cap. Um, in the region of maturation, the new cells have finished expanding, and now they begin to specialize into different types of root cells that have special functions. This is also called the region of differentiation. If you've never heard of the term differentiation before, um, we can talk about this. I think it's really easy to understand when we think about like a fertilized egg. Um, a fertilized egg of maybe a human being um, is just one cell type, right? It's you could say that maybe it's a stem cell, right? And it has to be able to differentiate into a huge number of different types of specialized cells before that fertilized egg or zygote or stem cell can become a human being. We've got brain cells and liver cells and muscle cells and lung cells and blood vessel cells and blood cells themselves and bone cells, and one cell type just is not enough to do all the functions that are required in a human body. So differentiation is the process of a more rudimentary cell type specializing into a mature special cell that has a designated function. One of the types of differentiation we see here is the development of root hairs, which are one of our first specialized cell types to develop. Um, Root hairs are single cells that have a very unusual shape. The root hair cells, they grow out from the epidermis of the root, and in doing so, they vastly increase the available surface area of the root for absorption of water, mineral nutrients, and oxygen. They are short-lived and they really only develop in the region of maturation. If you go back farther along the root to an older section of root, often the root hairs are gone. They have withered away and they're no longer present. So they're really short-lived. They're just present in that region of maturation, and the root hairs are doing the majority of the absorption of soil resources for that plant. Um, Root hairs particularly like to develop where a water-air boundary exists in the pore spaces of a substrate. So if you think about a potting mix or a soil, um, it's not all solid. A good potting media or a good soil is about 50% solid material. It's about 25% water and about 25% oxygen. There are spaces between the particles, and when there is a mix of both airspace and water in that pore, in the pore space in the substrate, that's where the root hairs like to develop. They don't develop very well if the media is too dry, because then there's not enough water. They don't develop well if the media is too wet, because there's not enough oxygen. And this is really important from a nursery and greenhouse management perspective, because the more root hairs that plant has, the better its growth will be. The better it will be at acquiring water and mineral nutrients and oxygen. So we both change the way we formulate potting media and we change the way that we water plants in order to uh, promote the development of root hairs. Um, Root decay is a major problem for horticultural producers in nurseries and greenhouses. It can also be a problem in landscapes where the soil is very degraded or where soil conditions are not optimal for the species of plant you're trying to grow. So in order to understand root decay, we first have to talk a little bit about just some basic cell physiology. Roots require oxygen, like all cells do, to perform cellular respiration. I shouldn't say all cells. There are some cells that don't do cellular respiration. They do things like fermentation instead. Um, but plant cells, plant roots require oxygen to perform cellular respiration. So when we look at that chemical equation, it's C6H12O6, which is glucose, and the plant oxidizes that with 6 oxygen molecules. And in oxidizing glucose, it's able to release energy, which it can use to do the work inside of the cell that the cell needs to do in order to live and perform the functions of life that are required to keep that tissue alive. Um, and then as a waste product, that plant produces 6 carbon dioxide and 6 water molecules. Um, because the roots of plants are essentially breathing out carbon dioxide, often in the pore spaces of a soil, there is a much higher concentration of carbon dioxide than in the air that we breathe above the soil. I think I want to say I've read stuff that said it was about 10 times higher concentrations of carbon dioxide in the soil compared to in the air. Um, so that's important to remember when you are thinking about the conditions that plants prefer underground. Um, there's a lot of ado made about overwatering and underwatering, but I want to kind of look at this as a more complex phenomenon. Um, there are a lot of ways in which root decay occurs, and they all have to do with inadequate oxygen in the substrate. So the first way that can happen is overwatering. The plant is just getting drowned with water. The substrate is completely flooded, and all of the pore spaces are filled with water. That excludes oxygen, and the root hairs die, and the root tissue can start to become damaged or infected due to the lack of oxygen. The second way that root decay can occur is if a plant is planted in a substrate that is inappropriately dense or fine grained. So if we look at this middle row of images um, on the left, there's an image that shows the difference in pore space between coarse-grained and fine-grained substrates. Coarse-grained substrates have much larger pore spaces, and that allows for better air, water boundaries. When you have really, really tiny pore spaces, and a really fine-grained substrate, all those granules of substrate pack together, and they really prevent there from being very much pore space. So plants don't tend to do as well in really fine-grained um, substrates. So that's another way, is an inappropriately fine substrate. And then the last condition that can be really harmful for plants are compacted substrates. So, for example, if during the construction of a house, the ground was run over with an excavator a bunch of times, the soil particles or the substrate particles themselves can become so compressed together that the pore space is inadequate, and water and oxygen can no longer flow freely through that compacted substrate. So there are at least three different factors which are affecting the likelihood that root decay occurs. Um, Overwatering, which is a decision we make, planting in a really fine-grained substrate that's not appropriate for that particular species of plant, or um, compacted substrates. Um, Without oxygen, those root hairs and the root tissues, they can't respire and they die. So when you look at plants which are experiencing root decay, you will not see those healthy white or yellow roots. You will see often kind of stringy brown roots which are dying. And the tissue's not healthy, it's not firm, it's not plump, it's not actively growing. Um, the fourth factor which can impact the probability of light decay are root decay organisms. Some of these are saprophytic, meaning that they are just present to decay already dead material. And if they're saprophytic root decay organisms, they're not really causing the problem. They're just feeding on the roots as a result of the suboptimal substrate conditions that we've created for a plant. Other root decay organisms are pathogenic, so they are disease-causing organisms that actively infect the roots and decay them and eat them. Um, Root decay organisms are much more prevalent, even the pathogenic ones, under overly wet conditions. Um, that can lead to seriously suboptimal plant growth and losses for greenhouse and nursery growers, and also in landscapes. If you look at this bottom image, you can see two Gerbera daisies. The one on the left has been grown with an appropriate watering, and you can see there's healthy, fine white roots growing through the potting substrate, and the leaves of the plant are quite large. It's a big plant. The plant on the right is a plant from the same crop of Gerbera daisies that was overwatered, and you can see that the leaves of the plant have not grown as much. They're smaller. There are fewer leaves, and we can't see any roots out to the edge of the root ball, because the roots continually are decaying. And if they're continually decaying, they cannot provide water and mineral nutrients from the substrate to the above ground portions of the plant. This all leads to me to question. Hopefully, it leads you to ask some questions too. How do aquatic plants survive? We have on Earth thousands of species of plants which live only in water, or which prefer to live in very wet, waterlogged soils. So, if these conditions, which are very prevalent in aquatic soils or substrates, are prevalent, how do aquatic plants survive basically under these overwatered conditions? And the answer is that they actually supply their own oxygen. They have piping internally inside of their roots to supply oxygen to those roots, even though they're growing in flooded, anaerobic mud where there's no oxygen. So the tissue that does this is called parenchyma. So parenchyma is this plant tissue. It contains very large spaces that allow the flow of oxygen throughout the internal tissues of a plant. This is pretty convenient. So if you think about a plant like a water lily, it has leaves on the surface, which are doing photosynthesis in the sunlight. And the leaf is interested in getting carbon dioxide out of the air to make sugars, and it's producing oxygen as a waste product, as part of photosynthesis. The oxygen that's a waste product from photosynthesis can be piped down the petiole of the leaf, so the stalk of the leaf, through the rhizome or underground stem of the water lily, which is in the mud, and then down into the roots of that water lily in order to supply the oxygen that that root needs in order to live and grow, even when there's no oxygen available in these flooded anaerobic soils. A lot of our terrestrial plants also have the ability to adaptively produce parenchyma. So this top image is a comparison of, on the left, a normal corn root, and on the right, the root of corn that developed under flooded soil conditions, stagnant anaerobic conditions where there was a lot of overwatering. So a lot of plants can actually change the way that they develop their roots in order to survive in suboptimal conditions, um, by producing parenchyma tissues in their roots. A lot of plants, though, can't do this. So we have to use special techniques in order to keep the roots of plants happy and actively absorbing mineral nutrients and water. And we do that through wet-dry cycles. So if you keep your potting media constantly saturated, that plant is not going to do well. It will experience root decay. If you keep your plant constantly dry, it's not going to do well. Its roots are going to desiccate, they're going to dehydrate, and the plant as a whole is going to dehydrate and die. What we want to do is alternate between a slightly higher level of moisture and a slightly lower level of moisture. And that wet dry cycle allows the plant to get a lot of water. And then as the media dries out a little bit, that allows oxygen into those pore spaces to keep the plant healthy. So, um, allowing for a wet dry cycle leads to really, really vastly improved plant growth. Um, when we look at this top image, this is a peat substrate moisture level scale. So there's level 1 to 5. I don't want to go too deep into what these mean. This is more of a greenhouse management question. But level 5 is saturated. The media should be shiny with water on the surface, and if you squeeze it, it will just actively drip water. In fact, you don't even have to squeeze level 5 substrate. It's just actively dripping. It's so wet. 4 is wet, and if you squeeze it, you can easily extract a lot of water. 3 is slightly drier, and you have to squeeze extremely hard to get out any water. 3 is the optimal place we want to keep most horticultural plants. Um, at level 2, there's still some residual water in the substrate, but it doesn't matter how hard you squeeze level 2 substrate, it will not release water. And then level 1 is almost completely dry. The peat starts to shrivel and shrink and crust up, and it'll actually pull away from the pot walls, kind of like a dried out sponge. Um, for most plant species, we would ideally, for virtually all horticultural species, we would love to stay permanently at a level 3. But it can be a little difficult to do that. So what a lot of growers do is they'll alternate between a 4 and a 2. They'll water to a 4, and then they'll allow the media to dry out to 2, and then they'll water to 4 again. And that ensures that that plant is spending, hopefully, the majority of its time at a level 3. Um, Looking now at not just how do we keep roots healthy enough to do absorption, but how do roots actually do the absorption of materials from the soil. Okay. There are two main types of transport, um, active transport and passive transport. And what I think is really important to notice is that plants have to be able to absorb water and dissolved mineral nutrients from the substrate, even if there's a very low concentration of those materials available in the substrate. So maybe a plant um, really needs calcium in order to survive, but it's growing in a very calcium poor soil. Um, if there's a really low concentration of calcium in the soil, lower than the concentration of calcium that the plant needs in its tissues in order to be healthy, the calcium is not really going to want to flow into the root of that plant because that would be flowing against a concentration gradient. Concentration gradients are basically a difference in concentration between one area and another. Typically, things like to flow from an area of high concentration to an area of low concentration. So if the plant's trying to stockpile calcium, but there's not much calcium in the soil, that plant is going to have to work in order to acquire that calcium. And we call that active transport. So active transport is when the roots use energy to actively absorb materials through the cell membrane, usually against a concentration gradient. Um, this often also occurs when certain molecules, plant nutrients, are too large to pass through the cell membrane unassisted. So when you look at this diagram of active transport, you can see that we're moving um, substances against the concentration gradient, and we're using energy in the form of ATP, adenosine triphosphate, which is the molecule that cells make to release energy inside of their cells, and they do that by breaking down glucose through the process of cellular respiration. So, if we look at the environment outside the cell, we can see that we've got three molecules or dissolved salt ions, and then we've got the cell membrane of a root cell. And we're just going to say that the outside of the cell part is the soil, right? So we have some dissolved mineral salts in the soil, and the plant wants to get the amount of nutrient that it needs in order to survive, even though there's not very much in the soil. So it's actually going to burn energy. It's going to use ATP to operate this little protein pump, which is purple in this diagram. It's going to operate this little protein pump that's going to attract these mineral salts using some electrochemical attractions. It's then going to transfer that into the cytoplasm or the goo inside the cell. Okay. Um, and it's going to do that over and over and over and over again. And that's how it's going to be able to stockpile nutrients that might be scarce in the soil. Many of a plant's mineral nutrients are actively absorbed. There's two reasons for this. One is that a lot of the molecules that plants are after, a lot of the minerals they're after, um, the mineral salts or the molecules are too large to pass through the cell membrane. And the other reason is because they're often scarce, and so the plant has to move them against a concentration gradient. Um, Passive transport's a little bit more simple. So in passive transport, the root cells don't have to use any energy to move materials across their cell membrane. Water typically just diffuses into the root tissue. So if you look at the first image in the passive transport section of the diagram, where it says diffusing particles, here we have some water molecules which are just able to slip through the cell membrane. Um, Obviously, the plant is constantly moving water up the stem and it's evaporating out of the leaves, and that process just draws more water in from the soil because the water wants to flow from an area of high concentration to an area of low concentration. When you look at this middle image, we have here a protein channel embedded in the membrane, but it's not a pump. It just sits there and it's open. And it allows for the free passage of maybe mineral salts through the membrane, even though those molecules might normally be too large to pass into the cell. And then We can also have carrier proteins that do this, that basically automatically change shape when they bind with the molecule, and that doesn't actually require the input of energy. It's just a passive process due to the chemical nature of those salts and of the carrier protein. Discussing how exactly these carrier and channel proteins work is really a subject for an undergraduate biochemistry class, and we don't have time in this lecture to explore that, but it is really, really interesting. It has to do with the chemical groups which are on these protein chains. It's all fascinating to me, but we don't have time to fully explore that. Just know that plants have to use energy to pull in most of their nutrients, and that most of the water is able to be absorbed passively, although there is some variability there. Okay. Um, when we look at active transport, um, this ends up relating to osmotic pressure within the root. So, osmosis, if you're not familiar, is a passive physical process, meaning passive meaning that it doesn't require any energy, physical meaning that this is just a feature of physics. This just occurs in any situation where water is moving across a semi-permeable membrane. So the membrane of a cell is semi-permeable. It allows some things in. It doesn't allow all things in. Um, and in osmosis, the water wants to move from an area of low solute concentration to an area of high solute concentration. Basically, the way I like to think about osmosis is the water wants to make sure that dissolved substances are all dissolved to the same concentration, regardless of what side of a membrane they're on. So if you look at this little diagram here on the right, you can see that in our first image, on one side of this U-shaped tube, we have a really low solute concentration. We have very few dissolved salts. And on the other side, we have a lot of dissolved salts. And what the water is going to want to do is it's going to want to flow from an area of low solute concentration to an area of high solute concentration so that everything is equally diluted. So water will actually flow through that semi-permeable membrane. It allows the passage of water, but it doesn't allow the passage of the salts. And it will equally dilute the the salts on both sides of that U-shaped tube. This is actually a pretty easy experiment to do at home with dialysis tubing. You can buy dialysis tubing online and you can cut it into little sections, and then you can tie it closed into little baggies. And if you fill a dialysis tube baggie with really concentrated salt water, and then you put it into a vessel, like a beaker containing distilled water, the dialysis baggie will the extra salts in the water inside the bag will pull water into the bag, and it will swell and sometimes burst. The opposite is also true. If you fill the dialysis bag with distilled water and then put it in really, really salty water, the water will flow out of the bag into the salt water in your beaker, trying to dilute it. So, um, when plants are actively transporting salts, dissolved mineral salts from the soil into their cells, they set up a difference in the solute concentration um, between the outside of the cell and the inside of the cell. So what happens next? We've got a higher concentration inside the cell. Where is the water going to want to go? Obviously, water doesn't have a brain. Water doesn't want anything. It's a an inanimate object, but the water is going to go somewhere as a result of having a higher concentration of salts inside the cell. The higher concentration of salts inside the cell that the plant is setting up by pulling in mineral nutrients is actually going to draw water into the root, and that actually creates a positive pressure which pushes water um, up the stem of the plant. Sometimes this can be so severe, especially in some smaller plants, that they have to have a way of venting that extra pressure to avoid bursting their cells. So if you look at this image in the bottom right corner, this is the leaf of a strawberry early in the morning. And those droplets of water, which are regularly spaced along the leaf, are not just some kind of magical photograph. Um, this is a phenomenon called guttation. So, in a well-nourished plant that is well hydrated, um, the active transport of uh, soil nutrients into the root can draw in so much water that the water pressure becomes so high they have to vent the pressure. So this is called guttation. Um, and the leaf has designated places at which to release this pressure as water droplets. Um, this is a normal process. You shouldn't be worried if guttation occurs. Usually it just means that the plants are well hydrated. It typically occurs at night. And the reason it occurs at night is the reason guttation occurs at night is because during the night, the temperatures are low and the plant's demand for water is also low. But it's also when the soil is cool and the plant's absorbing a lot of water to recover from the dehydration it experienced the day before. Um, so often the plant ends up kind of overhydrating itself. And then the next day, when things get hot and dry, that plant has basically a complete stockpile of water in its tissues. It'll partially dehydrate, and then the osmotic pressure will cause guttation again the next night. Um, in some species, osmotic pressure can actually be used to reverse embolisms in the xylem tissues of certain woody species. It's a more complex process than just root osmotic pressure. We don't have time to delve into that today, but it is possible for certain woody species to do that. Osmotic pressure, um, because it has to do with the amount of dissolved salts, um, our assumption here with on the last slide is that the soil nutrients are rare, that there's not a lot of mineral nutrient in the soil, but sometimes the level of dissolved salts is really, really high in the substrate. So we call this salinity. Salinity just describes the amount of dissolved salts in any kind of substrate. Um, when we have more salt in the substrate than the root has in its internal tissues, water is actually drawn out of the root. The water wants to flow from an area of low solute concentration to an area of high solute concentration. So if there's a lot of dissolved salt in the water in the soil, water in the root will want to move out of the root to dilute the salt in the soil. And that high salinity is very stressful for the plant. It can cause drought stress as water exits the root, and it can also lead to plant death in severe cases. There are a couple scenarios in which we see salinity causing um, osmotic stress. The first is over-fertilization. So I've got this little picture of some Miracle-Gro plant food. If you've ever used that blue plant food, you'll notice that it's crystalline. It is these tiny little crystals. They look like sugar or salt, and they are mineral salts. So they are the mineral salts that plants need in order to grow. Um, it usually is a combination of nitrogen, phosphorus, and potassium salts. And then there will also be maybe some calcium, magnesium, and sulfur salts, and probably some of the micronutrient salts as well. If you over fertilize your plants, you can cause drought stress or kill your plant because of the high salinity. That's why it's important to always make sure that you measure out those fertilizers appropriately. It's much better to slightly under fertilize than it is to over fertilize. Over fertilized plants have a lot of problems. I think it's much better to be lean on the fertilizer, um, and feed regularly, but with small amounts, small concentrations, rather than ever using high concentrations. The other scenario where we run into problems with salinity causing drought stress is sea salt. So in the ocean, there is a huge amount of dissolved NaCl, sodium chloride, which is table salt. Um, and sodium chloride is actually toxic to the vascular plants. So, animals and marine algae, things like kelp, which are not true plants, they evolved in the ocean and they use Na plus or the sodium ion from NaCl as an electrolyte. And electrolytes basically are dissolved salt ions that help an organism regulate the osmolarity of its cells, and they also help them regulate. So basically, they help the organism regulate how hydrated its tissues are. That's kind of what osmolarity often refers to in biology. And then the other issue is they can help send electrical signals between cells. Vascular plants did not evolve in the ocean, where there's a lot of sodium ions from the NaCl. Vascular plants evolved on land in freshwater ecosystems. So instead of using Na plus, they use H plus, the hydrogen ion, because that's relatively abundant in water, even freshwater. In freshwater, there's very low concentrations of NaCl, so they wouldn't really have the option to use Na plus. Um, so they use H plus instead. So table salt is actually toxic to the vascular plant, and it causes kind of severe drought stress. So um, plants have to have special ways of tolerating high salinity soils. Um, and there are very, very few plants that have developed those strategies. The high salinity of the ocean has actually prevented almost all the vascular plants from colonizing it. There are some notable exceptions. Um, Mangroves are a big one. The seagrasses, like Zostera and Posidonia, you get Zostera is known as eelgrass, and you get a lot of Zostera eelgrass beds along the coasts of Oregon, Washington, British Columbia, and Alaska. Posidonia is a tropical seagrass that grows in the Indian Ocean and around Australia. And in fact, there's a colony of Posidonia in a bay in Australia that's one of the largest vascular plants alive. Um, it's just one giant meadow that's all one plant. Um, and then there are also some species of beach grasses and some salt marsh species. This top right hand image is of an ice plant. Many of the species of ice plant are halophytes, meaning they're plants that are adapted to high salinity, and they grow in the wild in on beaches in South Africa, and they can tolerate really, really salty sand because they grow on a beach, and they have these succulent leaves, and succulents is a pretty common adaptation to be able to tolerate um, high salinity because the high salinity causes drought stress. A lot of these plants, like mangrove trees, seagrasses, beach grasses, and salt marsh species, they either have adaptations for drought tolerance, like succulent leaves, or they have an enhanced ability to tolerate and regulate the presence of salt in their tissues. Um, I'll bring up mangrove trees again later. Mangrove trees are actually using a variety of strategies. One of the ways that mangrove trees tolerate high salt conditions is actually by concentrating any of the salts they do take into their tissues into their oldest leaves. And then when those old leaves are senescing, once those old leaves are no longer, they've lived their useful lifespan, the mangrove trees will actually shed those old leaves and they're just packed full of salt. So um, that's the one way they can get rid of it in their old leaves. They can also prevent the salt from getting in, and also they have ways of regulating or preventing the loss of water from their roots to the surrounding salt. So but we'll talk about how that occurs later because it has to do with some internal anatomy of roots. Um, hmm. High salinity though is not. uh, I mean, it is a relatively frequent problem for plants, but in many ecosystems, um, dissolved mineral salts are scarce. A lot of plants are in fierce competition for the relatively small concentration of mineral salts there are in the soil, especially in something like a rainy forest. Forests are full of plants, they're all competing for nutrients, and the rain can dissolve those nutrients and wash them away or leach them down into the soil to a depth where plant roots can no longer reach them. So um, often there's pretty intense competition. So plants have to have different adaptations in their root systems in order to be able to compete. And we'll discuss a few of them coming up, including mycorrhizae. So um, you might think, wait, fungi aren't plants. Why are you bringing up fungi? And we have to understand a little bit about what fungi are before we can discuss mycorrhizae, because mycorrhizae are an adaptation that some plants, a partnership that some plants have developed in order to be more competitive for scarce mineral nutrients in competitive plant ecosystems like forests. So mycorrhizae are also called mycorrhizal fungi, and these are fungi which have a symbiotic relationship with plants. I'll talk about what makes their relationship symbiotic in just a minute. So when you go to the store and you buy some mushrooms, right? The mushroom is just the fruiting body of the fungus. It is not the main body of the fungus. It's just for reproduction. So um, a mushroom grows, it produces spores, the spores are released, and then the mushroom dies. But the main body of the fungus, called the mycelium, lives on. And the mycelium is much, much, much larger than the mushrooms. It's often 10, 100, 1,000, a billion times larger than the actual mushroom that was produced. And the mycelium is composed of many, many, many little threads called hyphae. And the hyphae are these cell thick filaments or threads of fungal tissue. Each one's just a single cell thick. And these little threads of fungal tissue just grow all over the substrate that the mushroom is living in, looking for organic material to decompose, from which the fungus can extract nutrients. So this image here is of button mushrooms. Button mushrooms are the popular type of mushroom that you can buy at the store, and they are grown in compost. So they're grown in a mixture usually of like wood or leaf material, and also maybe some composted food waste, or maybe some manure, and they decompose that organic matter in order to live. That's how they make their living. The really cool thing about mycorrhizae is they have partnered with they're species of fungi, typically mushrooms, which have partnered with plants. So mycorrhizal mycorrhizal fungi are able to decompose organic debris in the soil. For example, a rotting log. They can decompose leaf litter that falls every autumn. They can decompose the bodies of animals. They can decompose any kind of formerly living material. And in doing so, they collect the nutrients that were contained within that organic material. Um, this is where the relationship with plants comes in. The mycorrhizae actually form direct associations with the roots of the trees, and they do an exchange. The fungus, it really wants sugars, because sugars are food. There's no shortage of water and mineral nutrients in this forest floor, but there's not a ton of food for the fungus. So the tree will actually trade simple sugars produced through photosynthesis, glucose, in exchange for the mineral nutrient salts that the mycorrhizae acquired from the leaf litter, and also the water that the mycorrhizae are able to absorb. So often these cell thick fungal filaments have such a massive surface area. It's up to like 700 times the surface area of the actual root system of the plant. And so forming these associations with mycorrhizae actually helps these plants be much more drought tolerant as well and be much more successful in the long run um, in their ecosystems. Hmm. And there are a couple different kinds of mycorrhizae. I'm not going to go into all of the different species. That would be something that you could do an entire, career in mycology on. There are basically two different types. Most mycorrhizae fall into one of these two groups. There are arbuscular mycorrhizae and ectomycorrhizae. So arbuscular mycorrhizae are fungal species whose hyphae or their little filaments of their mycelium are actually able to invade the root cells themselves. So they actually puncture into the root cells, and within the root cells they develop these branching structures called arbuscules. And the arbuscules basically increase the surface area through which the mycorrhiza can exchange nutrients, waters, and sugars between itself and its host plant. Um, Approximately 78% of all living plants form relationships with these fungi, and it's a highly beneficial relationship for the both of them. Typically, there are some situations where these mycorrhizae can be just a little bit pathogenic, often mildly pathogenic. Sometimes, if the conditions are not great in the soil, the fungus will actually start stealing sugars without providing any any nutrients or water in exchange. But generally, these are beneficial relationships. Um, Ectomycorrhizae are a little bit different. They don't actually invade the cells themselves. They can't get into the cells. They instead form what's called a Hartig net, and that basically means they grow their hyphae or their little fungal filaments, which are just a single cell thick, into the root tissue, and they surround all of these root cells in order to exchange mineral nutrient salts, waters, and sugars with that plant. Plants have had a long time to figure out some of these nutrient problems and make partnerships with other organisms. Plants don't just partner with fungi to get mineral nutrients they need. They can also partner with bacteria. And in order to understand why a plant might want to associate with bacteria, we first have to understand a little bit about nitrogen. So, nitrogen is the mineral nutrient that's required by plants in the greatest quantities. There are a couple big problems with nitrogen. Um, First, I got to explain why it's important. So, nitrogen is an essential building block of amino acids. If you look in this top right-hand corner, you can see the um, kind of a basic diagram of an amino acid. You can see that there's an amino group on the amino acid. The side chart chain that's labeled R varies between different amino acids. And essentially, with these amino acids, they have to be strung together into long chains in order to make proteins. And the different side chains that are present on that amino acid are what give the protein its unique functions. But essentially, without nitrogen, plants can't make amino acids, and therefore they can't make proteins. If they can't make proteins, that means they can't build new cells. And if they can't build new cells, that means the plant can't grow. So nitrogen is super, super important. Some of the problems with nitrogen. Nitrogen is extremely abundant in the atmosphere. It makes up about 72% of the air we breathe. Super abundant in the atmosphere, but the nitrogen in the atmosphere is N2. It's tightly bound with a triple bond between those two nitrogen molecules, and it is unavailable for plants to use. Other problems that you face with nitrogen are that the water soluble forms of nitrogen leach out of the soil very, very easily. They move around and they're highly mobile. And that's not great if you're a plant and you want that nitrogen to remain available to you in that top layer of soil. Um, Fortunately for plants, there are some bacteria in the soil that can convert nitrogen gas in the atmosphere into water soluble forms of nitrogen, like ammonium, which is NH4, nitrites, which is NO2 minus, or nitrates, which is NO3 minus. And all of those can be absorbed by the roots of plants and used to produce amino acids and proteins and fuel new growth. Um, The bacteria that live in the soil, they're typically doing this for themselves. They need a reliable source of nitrogen if they can't get it from the organic material that they're decomposing. And so they have the ability to get it right out of the air and do the production of amino acids, make proteins, and then divide their cells and grow. Plants have learned to take advantage of this, though. And they do this by making the bacteria little houses. So lots of species of plants produce nodules on the roots, and these nodules are actually they exist to house these nitrogen-fixing bacteria. So the plant feeds the bacteria sugars from photosynthesis. So up in the canopy of the plant, in the leaves, it's producing sugars. Those sugars are flowing down through the phloem into the roots, and the plant is essentially farming the bacteria in these nodules on the roots by feeding the bacteria sugars. The bacteria want to live in the nodules because it's a lovely environment. There's a lot of food. They're protected, so it's a good deal. Um, the plants actually feed the bacteria so much sugar that the bacteria produce an excess of soluble nitrogen. The bacteria will end up producing far more water soluble nitrogen than they would ever be able to use for their own purposes. And so then the plant is able to take up that extra soluble nitrogen, and the plant can use that to build amino acids and proteins, and then, of course, new cells, and allows the plant to put on new growth. Plants that have bacterial nodules and form associations with these nitrogen fixing bacteria are known as nitrogen fixers because of their ability to fix atmospheric nitrogen converted into water soluble forms that are then available in the substrate for plant growth. There are a couple main groups of these bacteria. Um, I don't want to get too into the weeds on this because it's not the main point of this unit, but I do think that this is valuable to be aware of. Um, Rhizobium is a genus of nitrogen-fixing bacteria that makes associations with plants in the pea family, Fabaceae. So that would include things like peas, beans, and any other plants in the family as well, many of which are, shrubs and trees as well. A lot of farmers will plant species in the family Fabaceae to improve their soil fertility. So in the Midwest, for example, it's very common to plant soybeans and then alternate that with a crop of corn. So one year you grow soybeans, one year you grow corn. And one crop of soybeans not only produces all of the nitrogen that the soybeans need themselves to grow, the soybeans fix so much nitrogen in the soil that there's enough nitrogen for the subsequent crop of corn. So farmers are actually able to save a lot on their fertilizer bill if they alternate between growing soybeans and corn. They don't have to buy nitrogen fertilizers for their corn, which is a big deal because corn consumes more nitrogen fertilizer than any other crop. In the tropics, sometimes farmers will use fabaceous trees or leguminous. So these plants in the pea family are often called legumes. They will plant leguminous trees in rows through their fields, and these leguminous trees often have these feathery pinnate leaves that let a lot of light through, and they are also fixing nitrogen into the soil. So farmers will plant rows of these trees, and then they'll plant multiple rows of crops between each row of trees. And so the trees will slightly shade the crop and protect it from heat stress, but then also fix nitrogen in the soil to help it do well for things like coffee. There's another genus of bacteria that also forms associations with a couple groups of plants, and those are the Frankia bacteria. Frankia forms associations with trees in the oleaster family, Elaeagnaceae, and also with the alders. There are several different species of alders which are native to the Pacific Northwest and Idaho specifically. The red alder, Alnus rubra, the white alder, which is Alnus rhombifolia, and the gray alder, which is Alnus incana. And these alders are pioneer trees. So when there's a disturbance, like a forest fire or a landslide, the alders are often the first species to colonize those disturbed sites. And they grow extremely quickly, fueled by the nitrogen that they fix on the bacterial nodules on their roots. And they actually produce so much leaf litter and so much wood debris, and they fix so much nitrogen in the soil that they enrich the soil in those forest areas for the next several decades. Um, which means that even as those short-lived alder trees start to die and become replaced by conifers, they actually are providing the majority of the nitrogen that those conifers will be supplied with throughout their lives. Um, and then the last group of bacteria are the nitrogen-fixing cyanobacteria that form associations with the coralloid roots of cycads. So if you think about a cycad, like a sago palm, they have these really odd coralloid roots, and they're called coralloid roots because they kind of look like a coral from a coral reef. Um, and that's basically another type of nodule, that root nodule that a plant can use to feed a bacteria, house a bacteria, feed it sugars, and collect nitrogen for itself. One of the really cool things about these plants is that they're essentially self-fertilizing. So if you plant a tree that's in a pea family in your landscape, you're never going to have to supply that tree with nitrogen. That plant is not going to be capable of having a nitrogen deficiency because it hosts those bacteria in the nodules on its roots. If you plant an alder in a landscape, it can't have a nitrogen deficiency because it attracts these soil bacteria, feeds them sugars, and this basically helps grow itself. They're self-fertilizing, so that's a really good feature. Sometimes it can cause problems. If you look at this bottom left-hand corner, that is a Russian olive, and Russian olives are super invasive in North America, and they form associations with Frankia They're in the family Elaeagnaceae, and that allows them to survive on really poor soils and be a really horrible invasive species. So the more you know. Okay, getting back to some of our more basic root structure after our detour looking at nutrients. Um, Primary roots are the main central root of a plant. They're also called taproots. So if you look at this dandelion, that main central root that grows vertically downward, that is a taproot or a primary root. Secondary roots branch from the primary roots. These are also called lateral roots or side roots. We can keep going and we could say that the roots that branch from secondary roots are called tertiary roots. Roots that branch from those tertiary roots can be quaternary roots. We could say then quintenary roots, sextenary roots, septenary roots, octenary roots, nonary roots, and so on. But we don't need to do that. We just start saying, 10th order roots if you really care. Um, and unlike stems, which have nodes, roots lack nodes. So how do those lateral roots develop? Unfortunately, in order to answer this, we're going to have to look into more root anatomy. So we're changing now from longitudinal root anatomy to cross-sectional root anatomy. So rather than looking at the root lengthwise, we're looking at it crosswise. Um, when we look at stems of monocots and dicots, we might remember that monocots have vascular bundles which are scattered throughout the stem, and that dicots have vascular bundles which are arranged in a ring around the perimeter of the stem. Um, that pattern does not hold true for roots. So a lot of the same tissues are present in roots. We still have to have ways of getting water up the root to the stem and leaves, and we still have to have ways of moving sugars. So we still have xylem and phloem, but they are arranged in a different way. And we'll discuss the tissue organization within these roots as we go today. The first layer is the epidermis. We're going to start from the outside and move inward. The epidermis is the skin of the root. It's also sometimes called the epiblema or the pyliferous layer, and that's the outermost tissue layer of the epidermis, which produces the root hairs. Pyliferous means hair producing or hair bearing. The pili root word means hair. So if you've ever heard of depilation, like Nair, for example, Nair is a product that some people use to like remove hair from their legs. That's a depilatory agent. So D is not, against. Pili is hair. Ation turns a verb into a noun. So um, pyliferous means hair producing. That's the outermost tissue layer of the epidermis. I don't know that I have a lot more to add about the epidermis. It's the skin of the root, also called the epiblema or the pilophorous layer. The cortex is just under the epidermis. So it's a ground tissue. Ground tissues are any tissues which are neither vascular nor dermal, meaning they don't transport um, water or sugars or mineral nutrients, and they're also not the skin of the plant. Um, Compared to stems, which have cortex, there's a lot more cortex in a root. Um, the root cortex is mostly composed of loosely packed parenchyma cells. Parenchyma cells are these large, soft, thin-walled cells that do a lot of the metabolic processes in plants. So um, they are going to allow the flow of water and dissolved mineral salts to the vascular tissue at the center of the stem. Um, Unlike the vascular bundles in uh, stems, in roots, the vascular bundles are all concentrated together in the core of the stem called the stele. Um, the root cortex also often these cells will store a lot of the carbohydrates of a plant, and we'll look at examples of storage roots in a minute. Um, the exodermis is the outermost layer of the cortex, and it regulates the permeability of the root. So it kind of regulates what can or cannot enter the root. And then the endodermis is the innermost layer of the cortex, and it regulates permeability into the stele, which is the vascular core of the root. Um, and you might be wondering, how does that endodermis regulate permeability? How does it control what can or cannot enter the xylem of the root? In order to answer this question, we have to look at the ways in which materials flow through plant tissue. We've got two types. There's symplastic flow and apoplastic flow. So in symplastic flow, we have water and dissolved nutrients or other substances that are flowing through the cytoplasm of plant cells. If it's been a while since you've talked about cells, the cytoplasm is the solution inside of a cell that all of the chemical reactions are occurring in, that all of the organelles of the cell are present in. You can think of it as like the goo inside the cell. So symplastic flow is through the cytoplasm of the plant cells. And because it's occurring through the cytoplasm, the cell has some ability to regulate that flow. Plant cells are connected to each other through these openings in the cell walls called the plasmodesmata. And as symplastic flow passes from one cell to the next, it has to pass through the next cell's cell membranes, and they can control what can or cannot enter. So that's really valuable because not all dissolved substances in the soil are beneficial for the plant. Some of them are harmful, like salt. Um, Apoplastic flow is a little bit different. So apoplastic flow occurs through the cell wall itself. And because there are no cell membranes present to regulate it, plants can't control what's flowing through their tissues apoplastically. If you look at the top diagram, we can see this little orange arrow that shows apoplastic flow through the cell wall. You might be wondering, how does flow occur through the cell wall? Well, the cell wall itself is actually quite porous. If we look down at this bottom diagram, we can see the plasma membrane down at the bottom, which is yellow and green. The cell membrane, sorry, the cell wall, which is on the outside of the cell membrane, is composed mostly of these cellulose fibers, which are laid crosswise, almost like plywood, to increase their structural strength. But this matrix of uh, cellulose fibers is actually quite porous. You can see there's a lot of spaces through which water or dissolved minerals could flow. And can cause some problems for a plant which might be trying to keep harmful substances out of its tissues because they can't control what's flowing through that. The cell wall itself is not living tissue. It is mostly just these sheets of cellulose. And in order to stop apoplastic flow and have some control over what dissolved substances can enter the xylem and therefore be allowed to move up the stem of the plant and into the leaves, plants produce what's called a Casparian strip. This is a waxy, suberized, I'll explain what suberin is in a second, impermeable boundary layer which is secreted to prevent apoplastic flow through the endodermis in both directions, right? So when we look at this photograph, this microscope photograph in the top right-hand corner, you can see this kind of arching stripe that is wrapping around the stele or the vascular core of the root. And that is the Casparian strip. It's mostly composed of suberin, and suberin is this waxy substance that actually is what makes cork waterproof. It's why you can use pieces of cork bark to stop bottles of wine, and they don't leak, is because of the suberin, this waxy substance. And basically, they're packing this suberin into the cell wall itself, and that stops apoplastic flow and forces any water or minerals or dissolved substances into the cytoplasm if they want to move into the center of the root, into the xylem. This is important in both directions. First of all, it helps prevent harmful substances like salt from entering the xylem in excess. It also can prevent water, which the plant has in its vascular tissues, from backflowing out of the root if the soil becomes very, very dry. So the Casparian strip is a very important adaptation, both for preventing the entrance of undesirable materials and also for preventing the exit of desirable materials. The cells through which flow of water and dissolved substances is allowed, these cells are called passage cells, and they are able to regulate the flow of water and dissolved substances into the stele, which is the vascular core of the root, symplastically. So everything has to go through them in order to be allowed into or out of the root. As we've been discussing, the stele is the vascular core of the root. It's composed of several different types of tissues. We'll talk about these. There's pericycle, xylem, protoxylem, metaxylem, phloem, and pith. We typically only see a lot of pith in the roots of monocots, and it has to do with the way the vascular tissues are arranged within the roots of monocots. We'll talk about that in just a minute. So, starting, we've gone, just to review, we've started at the very outside, at the epidermis. Then we looked at the cortex, we looked at the exodermis, the cortex proper, and the endodermis, which helps regulate what substances absorbed through the epidermis and cortex are going to be allowed into the stele. Now we're starting with the outermost layer of the stele and working our way in. So we're going to start with the pericycle, which is the outermost layer of the stele. So the pericycle is the outermost tissue layer of the stele. It does it has two main functions. The first thing it can do is develop secondary roots. So if you look at this bottom left-hand image, this is a diagram showing the sequence of a developing lateral root from a willow primary root. So you can see the pericycle is this little ring around the center or core of the root. The root initially develops as this outgrowth of the pericycle, and then it progressively becomes larger and larger and larger until it erupts from the wall of the cell, literally tearing through the cortex and the epidermis to do so. This is a pretty interesting and almost violent process, and it's not at all like the way in which lateral branches develop from lateral buds, which are formed in the apical meristem. So secondary roots really are a product of secondary growth of the stem. There aren't like preformed buds in the roots as far as I'm aware. The pericycle is also somewhat oddly the source of secondary cambium in woody plant roots. And I say oddly because both the xylem and the phloem are typically to the inside of the pericycle. Nevertheless, the pericycle is a major contributor to the secondary cambium in woody plant cell roots, which allows them to thicken over time. Xylem transports water and mineral nutrients up the roots to the stem. Hopefully, this is a review. Um, in dicot roots, the xylem is usually arranged into arms. So if we look at this diagram of a dicot root, we can see that there are these four arms of xylem, which is shown in pink, which radiate out through the stele. And there's phloem between those arms. In monocot roots, the xylem and the phloem um, alternate, the tissues alternate to form this ring. So if you look at the monocot diagram, we have these pink bundles of xylem tissues and these green bundles of phloem tissues. In the dicot root, we have arms of xylem tissue, and then they're surrounded by phloem tissue. In woody plants, these layers end up getting united into a ring, and the growth of these woody roots often, they have growth rings just like a trunk, because they end up uniting these vascular bundles into solid rings. Roots often have two different types of xylem. They have protoxylem, which is composed of these really, really tiny small vessel elements, which are formed before the root elongates and matures. And then the metaxylem, which forms after the xylem vessels have matured. So or I should say, these are new tissues, new xylem tissues, which develop after the root has elongated. They develop in the region of maturation of the root. And you can see in this microscope slide, which has been stained with a variety of stains to show the different tissue types, we can see these really tiny protoxylem cells between the metaxylem and the phloem, and then we can see the large, well-developed metaxylem tissues internal to that. Phloem, I'm sure we're familiar with as well. This conducts sugars and other photosynthates, such as amino acids, which are produced in the leaves. It basically funnels them down to the roots. And phloem carries sugars produced by photosynthesis down the roots for two reasons. It fuels the growth of root tips, and it also, it fuels the growth of root tips basically by feeding those rapidly dividing cells in the root apical meristem and providing them with the fuel they need to grow and divide. It also allows for the storage of carbohydrates in the root cortex. So when we look at the cortex layer of the root, it's very, very thick. It's mostly composed of parenchyma. Parenchyma cells are these soft, thin-walled cells that are good at doing metabolic processes. So they're great for storing carbohydrates in. And many plants end up producing extraordinarily thick cortices. For example, these dahlia tuberous roots are a good example of a plant storing a large amount of carbohydrate in the cortex of the roots. There are a couple different major organizations of root systems that we see. We see tap roots, fibrous roots, and then a third type called adventitious roots. Tap roots are large, tapering, branched primary roots, and they're more prevalent in dicots. I know I've already mentioned primary and secondary roots earlier because I wanted to talk about how secondary roots form. But now I want to talk about some patterns in plant ID. Taproots, much more common in dicots. Monocots have root systems which are a little bit different. Monocots are much more likely to have fibrous root systems. So fibrous roots are roots with little to no taper and few secondary roots. So rather than being thick at the base and then tapering down to a tip, they kind of stay the same thickness the whole way down because most monocot roots are not capable of doing a lot of secondary growth. This top image you might recognize as onion. We have these unbranching fibrous roots developing from the basal plate of the onion bulb. This bottom image is of a palm. So palms also have a basal plate, and they basically form little root primordia from that basal plate, and they are continuously growing new roots. Because monocot roots are incapable of secondary growth, they only last so long before the vascular tissues break down, and then they have to grow more. So palms are actually constantly regrowing roots because their old roots are constantly dying and rotting away. Um, this is one of the reasons why certain types of palms blow over really easily during things like hurricanes. Um, you got to keep palms really, really healthy in order for them to have enough new, young, healthy roots in order for them to stay well anchored. Adventitious roots can be either taproots or fibrous roots. The important thing is that adventitious roots are roots which develop and grow from non-root tissues. So for example, when we see roots growing out of a stem, or when we see roots growing out of a leaf, those are adventitious roots. So in this top image, we have English ivy, and English ivy will produce roots directly out of its stem, and it uses them to root into the ground in its search for more nutrients, but it also uses them to climb up, to adhere its stem to tree trunks and brick walls to allow it to climb. In the bottom image, we have a leaf of a succulent, and it has formed rootlets at the base of the leaf, and it's also producing a new baby plant. These adventitious roots, we deal with them all the time in horticulture. Anytime you get a cutting to root, of any type, it's the cutting has to produce adventitious roots in order to um, survive. We deal with adventitious roots a lot in horticulture. Notice that it's adventitious, it's not advantageous, it's not adventurous. Adventitious is its own word that describes when plant organs develop and emerge from a dissimilar organ type. Roots can also have a lot of specialized function. I'll save us the time of reading this list and just get right into it. Many plants have storage taproots. These are used to store carbohydrates. Those could be sugars, simple sugars. They could be starches or polysaccharides, which are long chains of sugar molecules. If we look down at this bottom diagram, this is a diagram of a starch molecule. Unlike cellulose, which has bonds between the glucose rings, which alternate which side they're on, which helps keep the molecule straight. Starch molecules have all of their bonds between the glucose on the same side. These are called alpha bonds. So that causes these chains of sugar molecules to curl. And as they curl up, they kind of become tangled up into clumps, which form starch granules. And the cells in the cortex of these storage taproots will store these big starch granules. Um, Often, plants with storage taproots are biennial, which means they've got a 2-year life cycle. The first year, the plant produces just a rosette of leaves. So when you look at this top middle image, that's the rosette of leaves of a carrot. And that rosette of leaves is going to do photosynthesis. It's going to transport those down into the root and stockpile them. And it's just going to grow that root bigger and bigger and bigger and bigger and bigger, stockpiling those carbohydrate reserves. Then they'll go through a winter. The next year, they'll produce some leaves, and then the plant will bolt. And when plants bolt, what they're doing is they are rapidly consuming all of their stored carbohydrates to fuel the process of flowering and then to produce as many seeds as are possible. And there are a lot of our crop plants which will do this. So, like beets, carrots, parsnips, turnips, and radishes will all do this. It's important to harvest those storage taproots before they bolt, because once they've exhausted their carbohydrate reserves, they're no longer tasty and often they become quite unpleasant and woody. Tuberous roots. These are storage roots with at least some ability to grow adventitious shoots or stems from the proximal end. Remember, adventitious means that we've got a tissue growing from a dissimilar tissue type. So like if we look at a sweet potato vine, as it grows along the soil surface, anywhere the stem touches the soil, it can root in, and from those roots grow a sweet potato. The sweet potato is just a swollen storage root. And if you've ever left a sweet potato in your cupboard too long, you may have noticed that they will sprout stems and leaves from the proximal end. The proximal means closer to the center of the body of an organism. so not towards the ends or the extremities. So it's always the end that was closest to the stem that will sprout shoots. This top right-hand image is of some dahlia tuberous roots, and you can see the little green buds that are forming at the proximal end of those tuberous roots that will develop into the stems of a new dahlia plant. Water storage roots are used by a lot of plants which are native to really dry regions to stockpile water. And then because they've got a stockpile, they can actually continue to stay green and grow even during periods of drought. Um, you can usually tell if a plant is storing a lot of water because the roots won't be as solid. They'll be really juicy and often translucent. Um, One of the best examples of this are the spider plants. That's a very popular house plant. One of the reasons why spider plants make such a good house plant is that they have these um, thickened roots, these thickened transparent white roots, which they use to store a lot of water. So even if you forget to water your spider plant for a really long time, they have substantial water reserves in the soil or in the substrate, and they can draw on those to stay green and continue growing even if you neglect them. Daylilies. Hemerocallis are another group that has these thickened roots which store significant amounts of water. Stilt roots and buttress roots are easily confused, so I'd like to discuss them both on the same slide to talk about some of the differences. So stilt roots, these are aerial adventitious roots. So they form in the air, and they're adventitious, meaning they're growing from a non-root tissue, typically from the stem, and they're helping to stabilize that stem when a plant is growing in an unstable superstrate environment. So a superstrate is the opposite of a substrate. Super means above, sub means below. So, for example, if you are a corn stalk, this top left-hand image is the base of a stalk of corn. Corn will actually grow roots out of two, even three nodes on the stem above the ground, and it will grow those roots down to the soil as stilts. Those stilts help stabilize the stem, making it less likely to blow over in the wind, which is called lodging. Mangrove trees also produce stilt roots from their trunks, and these stilt roots help stabilize them on the beaches they grow on. Every day, the tide comes in and goes out twice. And when that happens, these trees get beat by the waves and the surf. So they have to be able to really stabilize themselves well in order to prevent their trunks from breaking. Buttress roots are a little bit different. They serve to stabilize the plant still, but they serve to stabilize the plant under kind of different forces. The buttress roots are more common to when the plant is trying to stabilize itself on an unstable substrate. So if you've got like a really soft, shallow soil forest, sorry, forest soil, which is on top of maybe a really shallow layer of bedrock, um, and the tree is experiencing a lot of wind, that tree could pretty easily blow over and just lift all the soft forest topsoil off of the underlying bedrock. And that is obviously bad for the tree. So a lot of trees will produce these buttress roots, which help stabilize them on basically soft, unstable soils or substrates. Um, Another good example are bald cypress. This bottom right-hand corner are the buttress roots of a cypress, but the bald cypress grows in swamps in the American Southwest where the soils are soft and boggy. And the tree in the bottom left-hand corner is the kapok, which is a um, tropical rainforest species with very famously fluted buttress roots. Contractile roots are really common in bulbs. So if you look at this large image on the right, these are the roots of a hyacinth, of a common hyacinth, and I've got a picture of the flower and leaves of that plant and also of its seeds on the slide as well. So looking at these roots, you can see that if we go to the tips, the roots are pretty normal. But then if we go back about halfway, the roots start to look all wrinkly. And that's because they've actually started to pleat and shorten. So contractile roots, they grow normally at first, but then once they mature, they start to pleat and shorten. And that helps pull these bulbous plants deeper into the substrate. And you might wonder, why would a bulb produce contractile roots. Why would it want to produce these roots which grow out into the soil and then pleat up, pulling it deeper? Consider that bulbs produce flowers and they're visited by pollinators, and those flowers are going to be pollinated, and the ovules of the fruits are going to be fertilized, and they're going to develop into seeds. Those seeds are going to fall in the soil somewhere, and they're going to germinate, and there's going to be a tiny little baby bulb growing on the soil surface. Bulbs don't want to be growing on the surface because the surface is a risky place. There's a lot of mammals and birds that might want to eat you. There are there's a lot more disturbance on the soil surface. The temperatures fluctuate more wildly on the soil surface. There's less water often on the soil surface. Often, if you can get down about 6 inches, the soil is more consistently moist. So these little baby bulbs have to have a way of digging themselves down, the 6 to 8 inches, sometimes even farther, that they would prefer to be to. So they use contractile roots to pull themselves deeper a little bit more every single growing season. Clinging roots are adventitious aerial roots that help plants adhere to some kind of support, such as a tree trunk or a cliff face. And the function of these is pretty obvious. If you are living in a highly competitive ecosystem like a forest, especially a tropical rainforest, there is a lot of competition for light. Light is not available on the forest floor because the trees are hogging all of it. So smaller herbaceous plants have to figure out a way to compete for light. And that might be, they could do that through evolving the ability to do photosynthesis under really low light conditions, or they could just choose to grow in a high light condition. So a lot of these plants actually choose to live on the body of another plant. We call them epiphytes. They use these clinging roots to adhere to the body of another plant. Some good examples could include vanilla orchid. This large image on the right is of a vanilla orchid, which is a vining orchid, which uses clinging roots to adhere itself to tree trunks to climb up into the canopy where there's a lot more sunlight. The more photosynthesis it can do, the more flowers it can produce, and the more seeds it will make. On the left-hand side, the top image is a branch in a tropical rainforest covered with bromeliads, which are popular houseplant. If we go one image down to this middle image, we have a Phalaenopsis orchid, which is growing on a tree trunk. You can see the roots, the clinging roots radiating out across that tree trunk to hold that orchid in place. And they're just happy to grow there. In a rainforest, they don't have to worry about drying out too much because it rains almost every day. And then this bottom left-hand image is actually the licorice fern, which is a very common and prevalent epiphyte in temperate rainforests in the states of Oregon and Washington, and also in British Columbia and southern Alaska. Lithophytes are plants that live on grow on rocks, and they're basically also using clinging roots in order to be able to do that. Propagative roots are roots which develop adventitious shoots. So we've talked about adventitious roots. We can also have adventitious shoots. So an adventitious root is developing from non-root tissue, and adventitious shoot is developing from non-shoot tissue. So these propagative roots, the plant grows roots out, and then the roots produce shoots far away from the main plant. And that helps the plant propagate itself and spread. This can be kind of a blessing and a curse. In a landscape, plants that do this tend to be viewed as weedy because they can pop up far away from where they're wanted. But it also makes them really easy to propagate. So if you want to propagate something like a trumpet vine or a quaking aspen, all you have to do is dig up some roots. I think it's best for them to be at least the thickness of a pencil and at least the length of a pencil. And basically dig up a section of root, cut it into pieces, pot it, and those pieces of root will produce new shoots. You can just dig up the pieces of root that already have shoots on them. My experience is that they tend to wilt a lot and they don't do well. I prefer to just use pieces of root, and that way they just produce a shoot when they're ready and they don't wilt so badly. There are some species which have decided that they don't need leaves and they can photosynthesize entirely with their roots. A really good example is the ghost orchid, which is native to the Florida Everglades in Everglades National Park. It only grows there in the wild and it has no leaves. This is an orchid which has, through the process of evolution, lost its leaves. It does all of its photosynthesis through this mass of green clinging roots by which it hangs on to the tree trunks it grows on. Trees are a particular interest in horticulture because they're foundational in our parks and gardens and in trees which produce, sorry, in nurseries which produce them. So there's a couple big important things to know about the roots of trees. The first thing is that roots of trees are almost all entirely within the top foot or two of soil. A lot of people are under the misconception that the roots of a tree form essentially the same shape as the top of the tree, but in reverse under the soil, and that's not the case. Even just a few feet down in the soil, there's almost no oxygen. Um, Often there's not very much water, and there's a not decaying organic material that's releasing a lot of nutrients. Often in the deeper layers of soil too, there's kind of a harsh chemical environment that a lot of plants don't really like. So most of the roots are typically pretty shallow, like I said, within the top foot or two of soil. That's where all the good stuff is: water, oxygen, and mineral nutrients from decomposing organic matter. If we imagine around the perimeter of the canopy of the tree a line drawn on the ground, we call that the drip line. This imaginary line around the tree visualizes a circle corresponding to the perimeter of the canopy. The drip line is extremely important in management of landscape trees. This is typically where you find the highest density of these fine feeder roots. The really thick, old, woody roots of trees are not good at absorbing mineral nutrients and water. It's those fine root tips in the region of maturation where we have root hairs um, that are good at doing absorption. So, because trees often have the highest density of little feeder roots at the drip line, this is usually the best place to apply fertilizers and other treatments to the roots of trees because it's where you get the best absorption. Um, Often in landscapes, landscape trees face significant disturbance. Trees' roots often go extend out from the trunk up to double or triple the diameter of the canopy. The root protection zone is kind of this imaginary zone. It's a guideline for preserving and protecting landscape trees. It extends out 2 times the canopy radius of an established tree. So basically, go from the trunk to the drip line, and then double that, and that's your root protection zone. Um, you want to avoid major disturbance of that zone in order to ensure that tree can live a long and healthy life. So, good examples of disturbance could be the installation of a driveway, the installation of a garage, any kind of major construction that's going to involve digging is going to damage the roots of that tree, introduce the possibility for decay organisms to get into the heartwood of the roots, and shorten the lifespan of that plant. Um, Another thing to consider is the less oxygen and water and mineral nutrients that the tree has access to as the area around it is paved, the less likely it is to do well. So, um, 2 very important ideas there: the drip line and the avoiding disturbance of the root protection zone helps your um, trees do a lot better in a landscape. And I really just loved this image on the top showing this. This is just a tree I imagine growing somewhere relatively wild. We've in the tropics. We've got this eroded soil profile, and we can see the different layers in the soil. It looks like we got a black layer there, which could have been a volcanic ash from an eruption, which soil has been deposited on top of since. But you can see that most of the roots of the tree are relatively shallow. They're growing outward. They're not going super deep into that soil profile. Pneumatophores are a really cool specialized type of root that you see in woody species like trees and shrubs, which are growing in anaerobic or flooded soils. So pneumo means lung or air, like pneumatics. And then the four part, I'm not, I don't remember what that means right now, actually. So these are roots especially for gas exchange. So basically, if you're a tree growing in these flooded anaerobic soils, you need a way to get oxygen to your roots. And maybe because you're a woody plant, you don't have a lot of parenchyma tissues, those spongy air conducting tissues in your woody trunk, because that would make your trunk weak and soft and you would fall over. So you have to instead have a more direct way of getting oxygen to your roots. You can't send it down from as a waste product from your leaves. So um, these species produce pneumatophores or knees. So if you look at this top image, this is a black mangrove, and we can actually figure out where its main roots are based on the lines of pneumatophores, or these little air-breathing roots, these snorkel roots almost, that are popping up out of the mud on this tidal flat. Um, the bottom image is of a bald cypress. Bald cypresses are native to freshwater swamps in the American Southeast, and they can actually be grown very far north of their native range. If you visit Ann Morrison Park, you can actually find a lot of these trees growing around the ponds. And the trees around the ponds have a lot of pneumatophores. And the reason they have a lot of pneumatophores is the water level in the pond roughly corresponds to the water level in the surrounding soil. Those soils are flooded just a few feet down. So the tree has to have a way of supplying oxygen to those flooded roots. And the solution are these little snorkel roots, which allow for the intake of oxygen, and then they're able to send that oxygen down through the root through some parenchyma tissues to keep the root tissues alive and growing. um, so not all plants use their roots for good. Some plants are evil and they're parasites. So parasites, in case you need a refresher, they're organisms that feed off of a host organism without killing it. The parasite benefits and the host is harmed. So Haustoria are these modified roots which parasitic plants use to siphon water, mineral nutrients, and sugars from the stems, roots, or leaves of their host. So if we look at this top image, you can see that this is a cross section of a branch of a tree, and then we have this different patch of yellow wood with a big knob on the top, and that is the haustorium of a mistletoe. So mistletoe, when it germinates, it actually produces a radical or a little embryonic root that plugs into the stem of the tree. And then it starts siphoning off water and mineral nutrients and fueling its own growth. This haustorium invades the branch to such an extent that you can see the branch is much thicker before the haustorium, and then it's much thinner after the haustorium. The mistletoe is actually drawing off really large amounts of water and mineral nutrients, and that actually reduces the growth of the branch farther down. If you are familiar with, if this middle picture looks familiar to you, this is Rafflesia arnoldii. It is famous for having the largest single flower of any species of plant. The plant is patterned to look like a rotting carcass to a fly, and it also smells of rotting meat. And so it's pollinated by carrion flies in these rainforests in Southeast Asia. Rafflesia arnoldii actually has no leaves and no above ground stems. It has a rhizome under the soil that has haustoria plugged into the roots of certain species of vines. And these vines are capable of growing up into the forest canopy and getting lots of light. And Rafflesia lives entirely underground and just steals sugar, water, and nutrients from its host. Rafflesia has actually been a parasite for so long that the species has lost the ability to produce. Not only does it not produce leaves, it has lost the genes to be able to produce the green pigment chlorophyll in order to be able to do photosynthesis, which I think is fascinating. And then this bottom image is a parasitic plant that's common in Idaho called dodder. And daughter produces numerous adventitious haustoria right out of the stem, and it plugs into any vascular bundle it can get a hold of, either a stem or a leaf. You can see in this image the piece of stem has actually been ripped off of the leaf, and you can see the little spots in the leaf, the scars where the haustorium were plugged in to siphon off um, water and mineral nutrients. Um, and then I think this is our last content slide. Orchids have really, really unusual roots, and that makes them kind of hard to grow. So tropical orchids, super, super popular. They've got these colorful flowers, unusually shaped flowers. That makes them really popular houseplants. A lot of people kill orchids because the roots are highly specialized. So if we think back to the slide where we talked about epiphytes, plants which grow on the bodies of other plants, and I showed an image of an orchid growing on the trunk of a tree. Orchids have to have adaptations to be able to do that. And the adaptation that orchids have is called a velamen. So there's this thick gray spongy layer covering the steel or the core of the root um, called a velamen. And that helps absorb water, and it also adheres orchids to whatever substrate they're growing on. So if you look at these orchids, you can see that the healthy roots are these kind of greenish gray roots. Um, the velamen essentially is so absorbent, the orchid doesn't really need potting media. It doesn't need the potting media to be able to absorb a lot of water, because if the orchid roots get wet, they actually absorb water, and then from there, the core of the root can draw water out. So they already have the ability to absorb water just in the outer covering of the root, the velamen. The problem people run into with orchids as houseplants is when they pot them in a regular potting media, or if they overwater the orchid, or if they leave it sitting in standing water, that velamen is spongy and absorbent, but it's also composed of dead cells. So if you have excess of moisture and insufficient airflow, it will just rot away. Um, and once the velamen has rotted away, the orchid doesn't really have any good ability to absorb water. That living inner core dies if the velamen rots away. So it's super important to grow orchids in special potting media that's formulated for orchids. Typically, it's made of things like fir bark or pine bark, or some types of specialized fibers, or certain types of moss. You don't want to grow them in regular potting mix, and you want to make sure that you get the velamen wet when you water them, but you don't want to leave them sitting in a moist substrate. All right, so that brings us to the end of our presentation. I just wanted to drive home, just because we overlook roots often, doesn't mean you have to keep overlooking them. Just because we don't normally see them doesn't mean they're not important. Roots do a lot of really important things that I wasn't able to talk about today too, like prevent erosion. They also contribute to the formation and consolidation of soils. They help hold all the soil on our planet together so that the earth doesn't turn into one giant dust bowl. Um, they also help break up ground rock, bedrock, and weather parent material in order to make new soils. Roots acquire all of the raw materials to fuel the beautiful stems and leaves and flowers and fruits that we grow plants for. They anchor plants, they store food for plants, many of many of those roots which store food are delicious. And they also perform a wide range of special functions which help plants survive in really, really challenging environments. So overall, I think roots are underappreciated. They're doing some really, really fascinating things underground. They're just a little bit harder to look at and study because they're often hidden from us. Um, I hope you enjoyed this. I hope it was informative, and I hope you have an excellent day.